Hello, welcome to Biology, the scientific study of life. Six linked genes exhibit unique patterns of inheritance. We begin by studying the chromosomal basis of sex determination in humans and some other mammals or animals. The chromosomal basis of sex, whether we are male or female, is one of our more obvious phenotypic characters, although the anatomical and physiological differences between women and men are numerous. The chromosomal basis for determining sex is rather simple. In humans and other mammals, there are two varieties of sex chromosomes designated X and Y. A person who inherits two X chromosomes, one from each parent, usually develops as a female. A male develops from a zygote containing one X chromosome and one Y chromosome. The Y chromosome is much smaller than X chromosome, as we see in this figure. And only relatively short segments at either end of the Y chromosome are homologs with the corresponding regions of the X. These homologous regions allow the X and Y chromosome in males to pair and behave like homologous chromosomes during meiosis in the testes. In both testes and ovaries, the two sex chromosomes segregate during meiosis, and each gamete receives one. Each ovum contains one X chromosome. In contrast, sperm fall into two categories. Half the sperm cells a male produces contain an X chromosome, and half contain a Y chromosome. We can trace the sex of each offspring to the moment of conception. If a sperm cell bearing an X chromosome happens to fertilize an ovum, the zygote is XX, a female. If a sperm cell containing a Y chromosome fertilizes an ovum, the zygote is XY, a male. This sex determination is a matter of chance, a 50-50 chance. Besides the mammalian XY system, there are three other chromosomal systems for determining sex are shown in this figure, as we see here. In parts BD, as we see, notice that humans, the anatomical signs of sex begins to emerge when the embryo is about 2 months old. Before then, the rudiments of the gonads are generic. They can develop into either ovaries or testes, depending on hormonal conditions within the embryo. Which of these two possibilities occurs depends on whether or not a Y chromosome is present. In 1990, a British research team identified a gene on the Y chromosome required for the development of tests. They named the gene SRY for sex determining region of Y. In the absence of SRY, the gonads develop into ovaries. The researchers emphasized that the presence or absence of SRY is just a trigger. The biochemical, physiological, and anatomical features that distinguish males and females are complex, and many genes are involved in their development. SRY codes for a protein that regulates other genes. Researchers have subsequently identified a number of additional genes on the Y chromosome that are required for normal tests functioning. In the absence of these genes, an XY individual in male but does not produce normal sperm. Inheritance of sex-linked genes. In addition to their role in determining sex, the sex chromosome, especially X chromosomes, have genes for many characters unrelated to sex. A gene located on either sex chromosome is called a sex-linked gene. Although in humans the term has historical referred specifically to a gene or on the X chromosome. Note that distinction between the term sex-linked gene referring to a gene on a sex chromosome and linked genes referring to genes on the same chromosome that tend to be inherited together. Sex-linked genes in humans follow the same pattern of inheritance that Morgan observed for the eye color locus in Drosophila. Fathers pass sex-linked alleles to all of their daughters, but to none of their sons. In contrast, mothers can pass sex-linked alleles to their sons, to both sons and daughters. A sex-linked traits is due to a recessive allele. A female will express the phenotype only if she is a homozygote, because males have only one locus. The terms homozygous and heterozygous lack meaning for describing their sex-linked genes. The term hemizygous is used in such cases. Any male receiving the recessive allele from his mother will express the trait. For the reason, for more males than females have sex-linked recessive disorders. However, even though the chance of a male inheriting a double dose of the mutant allele is much less than the probability of a male inheriting a single dose, there are females with sex-linked disorders. For instance, color blindness. Daughter may be born to a colorblind father whose mate is a carrier. However, because the sex-linked allele for color blindness is relatively rare, the probability that such a man and woman will mate is low. A number of human sex-linked disorders are much more serious than color blindness. An example, Duchenne muscular dystrophy, which affects about 1 out of every 3,500 males born in the United States. The disease is characterized by a progressive weakening of the muscle and loss of coordination. Affected individuals rarely live past their early 20s. Researchers have traced the disorder to the absence of a key muscle protein called dystrophin and have mapped the gene for this protein to a specific locus on the X chromosome. Hemophilia is a sex-linked recessive disorder defined by the absence of one or more of the proteins required for blood clotting. When a person with a hemophilia is injured, bleeding is prolonged because a firm clot is slow to form. Small cuts in the skin are usually not a problem, but bleeding in the muscles or joints can be painful and can lead to serious damage. Today, people with hemophilia are treated as needed with intravenous injection of the missing protein. X inactivation in female mammals. Although female mammals, including humans, inherit 2 X chromosomes, 1 X chromosome in each cell becomes almost completely inactivated during embryonic development. As a result, the cells of females and males have the same effective dose, 1 copy of genes with loci on the X chromosome. The inactive X in each cell of a female condenses into a compact object called a parabody, which lies along the inside of the nuclear envelope. Most of the genes of the X chromosome that forms the parabody are not expressed in the Notice this picture, two cell populations in adult cat. Okay, back to parabody. Chromosomes are reactivated in the cells that give rise to ova. So every female gamete has an active X. British geneticists Mary Lyon demonstrated that selection of which X chromosome will form the parabody occurs randomly and independently in each embryonic cell present at the time of X inactivation. As a consequence, females consist of a mosaic of two types of cells. Those with the active X derived from the father, and those with the active X derived from the mother. After an X chromosome is inactivated in a particular cell, all mitotic descendant of that cell have the same inactive X. Thus, if a female is heterozygous for a sex-linked trait, about half her cells will express one allele, while the other will express the alternate allele. The others will express the all. Notice this figure shows how this mosaicism results in the mottled coloration of tortoiseshell cat. In humans, mosaicism can be observed in a recessive X-linked mutation that prevents the development of sweat glands. A woman who is heterozygous for this trait has patches of normal skin and patches of skin lacking sweat glands. Inactivation of an X chromosome involves modification of the DNA, such as attachment of methyl groups SH3 to one of the nitrogenous bases of DNA nucleotides. The regulatory role of DNA methylation. Researchers also have discovered a gene called XIST for X inactive specific transcript that is active only on the parabody chromosome. Multiple copies of the RNA molecule produced from this gene apparently attached to the X chromosome on which they are made, eventually almost covering its interaction of this RNA with the chromosome seems to initiate X inactivation. Our understanding of X inactivation is still rudimentary, however. Back to X inactivation and the tortoiseshell cat. Notice that. The tortoiseshell gene is on the X chromosome, and the tortoiseshell phenotype requires the presence of two different alleles, one for orange fur and one for black fur. Normally, one females can have both alleles because only they have two X chromosomes. If a female is heterozygous for the tortoiseshell gene, she's tortoiseshell orange patches are formed by populations of cells in which the X chromosome with the orange allele is active. Black bitches have cells in which the X chromosome with a black allele is active. Notice that. Calico cats also have white areas, which are determined by yet another gene. Okay, thanks for listening.